Hi everyone, and welcome back to STEM Revise. Today we're diving into carbohydrates, specifically monosaccharides, disaccharides, and polysaccharides. These are essential biological molecules acting as the main energy source for living organisms and key players in cellular structure. We'll break down each type from simple sugars to complex carbohydrates and make sure you understand their structures, functions, and how they connect to A-level biology. So let's jump right in. Let's start with monosaccharides. These are the simplest unit of carbohydrates, a single monomer of sugar. Monosaccharides are small, soluble, and sweet-tasting molecules that provide quick energy. Glucose, galactose, and fructose are all examples of these, and each of them have the same chemical formula, C6H12O6, but they differ in structure, making them isomers. These slightly different structures lead to unique properties, which is why glucose, for example, is the primary energy source in cells. When two monosaccharides combine, they form a disaccharide. This process, known as a condensation reaction, as we discussed in the previous video, links the two molecules together, producing a molecule of water as a byproduct. Common disaccharides include sucrose, lactose, and maltose. Sucrose, for example, is made from glucose and fructose, while lactose is made from glucose and galactose. Disaccharides are still relatively simple sugars, and they're often used for energy, just like monosaccharides. When three or more monosaccharides link together, they form polysaccharides. These large molecules are built from many monosaccharide units, making them complex and ideal for storage. For example, in plants, glucose units link to form starch, which stores energy. In animals, we find glycogen, which stores glucose in our liver and muscles. Due to their size, polysaccharides are insoluble, which means they don't dissolve in water, a perfect quality for storage molecules. Plus, because they're so large, they don't affect the osmotic balance of cells, making them efficient and compact energy reserves. So to summarize this slide, monosaccharides are quick energy sources. Disaccharides are formed from two linked monosaccharides, and polysaccharides are complex storage molecules. Each type plays a critical role in biological systems, supporting everything from immediate energy needs to long-term energy storage. When two monosaccharides combine, they form a disaccharide through a reaction called a condensation reaction. This process is important because it links the two sugar molecules together by forming a glycosidic bond, with water released as a byproduct. Let's take a look at some examples. So when two glucose molecules join together, they create maltose, which is often found in germinating seeds as a source of energy. If glucose pairs with fructose, we get sucrose, which is a common table sugar and the main transport sugar in plants. Finally, when glucose combines with galactose, it forms lactose, the sugar found in milk. During each of these reactions, a water molecule is removed. This removal allows the monosaccharide to bond through a covalent glycosidic bond, resulting in a disaccharide. This bond formation is crucial for building larger carbohydrate molecules and for storing energy in forms that organisms can later break down as needed. Monosaccharides and some disaccharides, like maltose, are reducing sugars. This means they can donate electrons to another substance, essentially reducing it. This property is useful in lab tests, where reducing sugars can react with chemicals like the Benedict solution. Benedict's test helps us detect the presence of these sugars by causing a color change. To perform this test, here are the steps. First, we grind up the food sample that we want to test in water to release any sugars. We then add 2 centimeters cubed of the sample to a test tube. We then add 2 centimeters cubed of Benedict's reagent. And then finally, we heat the mixture gently in a boiling water bath for around 5 minutes. If reducing sugars are present, the solution will change color from blue to green. yellow, orange, or red, depending on the amount of sugar. This reaction confirms the presence of reducing sugars within a food sample. So, unlike reducing sugars, sucrose is a non-reducing sugar, meaning it doesn't change the colour of Benedict's reagent when heated with it. To test for a non-reducing sugar, we first confirm that no reducing sugars are present, and we then break down the sugar to see if a reaction occurs. Here's how the test works. First, we grind up the food sample in water. We then add 2 centimeters cubed of the sample and 2 centimeters cubed of the Benedict's reagent to a test tube. Then we place the test tube in a boiling water bath for 5 minutes. If it stays blue, we know that a reducing sugar is not present. Then we take another 2 centimeters cubed of the food sample and add 2 centimeters cubed of dilute hydrochloric acid to break down any non-reducing sugars. We then place this mixture in a boiling water bath and slowly add sodium hydrogen carbonate to neutralize the acid, the hydrochloric acid. And then finally, we retest the solution again by adding Benedict's reagent and heating. If a non-reducing sugar, like sucrose, was present, it should now turn an orangey brown sort of colour. This test allows us to confirm the presence of non-reducing sugars after breaking them down into reducing sugars. So to summarise, in this video we covered the basics of carbohydrates, starting with the definitions for monosaccharides, disaccharides and polysaccharides. We then went on to look at how to test for reducing sugars using the Benedict's test and how to detect non-reducing sugars by first breaking them down with acid and then testing again. These methods are essential tools for identifying different types of sugars in biological samples.